This video is going to deal with the chemical part of this section. It's not very long. The physical really only did two things. We did two examples of moist and dry heat experiments. Now, there are a large number of chemicals, and there are chemicals that are sterilizing chemicals. We accidentally ordered something called Bacterial one time. The box had teeth, like a smile with teeth coming out of it. You had to clear the building. If you used it, you had to wear like a hazmat suit and gloves, and so that obviously was too much. But there are two categories of chemicals. Now, a disinfectant and a chemotherapeutic agent. You can think of that as an antiseptic in a way. Chemotherapeutic, meaning literally you're taking drugs to kill disease. But here are some terms. An antibiotic is a term, it's a chemical produced by microorganisms that kill other microorganisms or organisms. That's the definition. Anti is without, bio is life. Static, now this is a drug example. The term static in regards to bacterial growth, there are some drugs that are called bacterial static drugs. They prevent the infection from getting worse, because a person might have such a huge infection that if you were to give them some sort of medication that would kill all the organisms at once, it would make them actually more sick, because you get lysing of cells and toxins. So a static situation is where you allow a person's immune system to catch up and take out the infection naturally. Don't really know much about that as far as cleaners go. The word cidal means to kill, so if you see it on the end of a word, it's a suffix like bactericidal, fungicidal. Not to poke fun, but you think about homicidal, if you joke around. I'm feeling homicidal, which is not a good joke in our time. But cidal means to kill. You know, homo sapiens, virucidal, fungicidal. Disinfectant, covered this earlier. It's used on inanimate objects, tables, chairs, floors, anything not living. And an antiseptic is a chemical used on living tissue. If you spray rubbing alcohol or alcohol that's watered down on a table, it's being used as a disinfectant. If you spray it on skin or rub it on skin, it's being used as an antiseptic. So here are some things that are tricky to kill. We talked about bacillus with the physical control, Clostridium tetani, Clostridium botulinum. Those are the bad bacteria. But tuberculosis, waxy coat, anything that forms a spore, fungal spores, they float in the air. The hepatitis virus has an envelope. It's hard to kill. Pseudomonas is a bacteria. It's actually called Pseudomonas aeruginosa. And extremely resistant, can grow in betadine. It's pretty prominent in people who have third-degree burns. They get infections of that. Okay, factors that affect physical can also affect the chemical. pH, time in contact with the microorganisms, what type of method. A disinfectant, which again is used on inanimate objects. If you're talking about tissues, you're talking about an antiseptic being used. But skin has got oil on it, it's got folds, it has dirt involved, so treating skin can be tricky as well. Now, here's a lab exercise, and I'll try to make this as painless as possible. Clearly, we're not going to be grabbing tubes, but just go with me on this. What we do in the lab is you'll have 4 tubes of sterile broth for each organism. So the group, and it says per pair, so you have 4 people at a table. So you're going to need 8 tubes. You have 2 cultures of E. coli and bacillus. E. coli is a gram-negative, does not form spores. Bacillus is a spore former, a little harder to kill. If you have an old culture, they form spores, so they're a lot harder to kill. And then some kind of disinfectant that you're going to use. So carefully pour the pre-measured amount of a disinfectant into each of your cultures. Now, so if you have an E. coli broth, a liquid culture, and sorry to bounce back and forth, I'm going to go way back up here. Here is a culture with some growth in it. So you have something like this. Maybe it's got a little bit more broth, but you got some E. coli in a broth culture. So what you would do is figuring out what kind of disinfectant to use, and that was always kind of fun. You would then pour the disinfectant into the culture, and then you start a timer. This whole thing is going to take 15 minutes. So 2 people will be doing E. coli, 2 would be doing bacillus. The whole entire experiment takes 15 minutes. And then, of course, you want to label everything so you can find it, and then you're going to incubate it for 24 hours when you're done with all of your assignment. So what would you be doing exactly? At intervals 2, 5, 10, and 15. Okay, the whole thing's going to take 15 minutes, but you're going to be busy from the time you added your disinfectant until the 15-minute mark. So here's what you're going to be doing, or you would be doing. See there, it says culture plus disinfectant. So you're going to have some E. coli. Let's say you pour some Clorox into there, a certain amount, and it fills it all the way up to the top. You set the culture down. Like I said, you start a timer. At 2 minutes, you would take a couple of loopfuls out of this culture plus disinfectant, and you would transfer that to a sterile broth, and you would label that broth as a 2 minute. That broth represents you're getting cells that have sat with the disinfectant for 2 minutes. And you're going to put it in there. And what I tried to draw here was some sort of growth, but to give you an idea if there's any growth at all. But you would put it in there, and then you're going to wait. Now, at the 5-minute mark, so you wait basically 3 minutes, you go back to the original culture plus disinfectant. You take a couple of loopfuls, and you transfer that to the 5-minute broth. Okay, at the 10-minute mark, the whole thing's going to take 15 minutes. You're going to go back to this again, and if you follow this up, see it goes all the way to there. You're going to take another couple of loopfuls. You're going to put it in here, and you do aseptic technique the entire time. You don't want to contaminate it. But the 10-minute broth is getting cells that have sat with the disinfectant for 10 minutes. And then you're going to do the last one at the 15-minute mark. So you take some cells from here, and you're going to put them in another sterile broth, and they've sat with the culture for 15 minutes. And what you're doing here is you're going to be able to measure out, and then you would then incubate these. You would come back and look for growth. Did you get any growth in any tubes? The culture plus disinfectant, you throw that away. It's gone. What this does is kind of cool. It'll give you a time frame. It'll tell you how long did it take that disinfectant to start killing cells. If you have growth in the 2-minute and growth in the 5-minute, and then the 10 and 15 have no growth, that means it took at least 5 minutes for the chemical to start working. If you have growth in all 4 tubes, that's not a very good disinfectant. It means even after 15 minutes, it hadn't killed all the cells. If you have no growth in the tubes, that's a very good disinfectant, which Clorox is. It also kills the coronavirus. But so anyway, so that's how you look at these. So that's going to be our lab scenario. So once you would inoculate these tubes, you would then put them in an incubator for 24 to 48 hours, and you come back and read the results. So let's do this one first. Rubbing alcohol. Done this in the past. Here is the series of E. coli tubes after they've been incubated. And here's the series of bacillus tubes after they have been incubated. And then here were the original culture, just so you can get the idea. So what you would do in these tables were blank. You would check for growth, no growth. That's really all you can tell with a broth tube. You would shake them around. If it's cloudy, if you see things inside, that was growth. If it wasn't cloudy, like see how clear this one is compared to this one, this is a no growth. And it's all or none. You don't look at it and go, oh my gosh, I see bubbles. That has nothing to do with it. It's either going to have growth or it's not. So rubbing alcohol is a pretty common result. These were the results. The 2, the 5, and the 10-minute tubes had growth, and the 15 often would not have growth. And that's pretty common for alcohol. Alcohol takes 15 minutes to start working, usually. Bacillus is not killed by alcohol. It's a spore former, hard to kill. You can see there's growth in all 4 of the tubes. So if somebody said, hey, I have some bacillus in my house, I'm going to treat it with rubbing alcohol, you would say that's not a good idea. It's not going to do anything. Which you really, spores are hard to kill anyway, but you're really just going to be wiping them off. Clorox, another common result. So basically you would add your detergent to your culture. That's what that is. See the growth in there. Here's what Clorox does. See these 4 clear tubes? This is the E. coli ones. And then here's the bacillus. And that's pretty common result. Sometimes the bacillus would have growth, but Clorox wipes out just about anything. So there was no growth in any of the tubes. I mean, even immediately. And if you ever do this experiment in a lab, when you pour Clorox in a tube, it actually starts heating up. I mean, it's that strong. So Lysol, see the bottle? Pretty common result. The first 2 tubes of E. coli had growth. The last 2 did not. So it takes a while for Lysol to start working. Yeah, and it is kind of a different setting. I mean, you usually wipe off things with Lysol. So Bacillus was not usually killed by Lysol. It is a spore former, but you are wiping it off. You're at least doing something. You're clearing off the Bacillus, but that was a pretty common result. Betadine, which is what's used for skin. Now that it is an antiseptic, betadine usually did not have good results either way. There was growth in all the tubes, but this is not a setting with skin involved. Skin's acidic, and betadine is a pretty strong, it is a good cleaner. It's not going to kill a spore former. Sometimes it did kill some of the E. coli, but a lot of times you had growth in all the tubes. And Aunt Fanny's cleaning vinegar. So vinegar is acidic. If you have something that's like a pool, like a swimming pool machine that gets chlorine deposits on it, if you soak it in vinegar for like an hour, vinegar will dissolve the chlorine. So the E. coli tubes with the Aunt Fanny's cleaning vinegar, first 3 had growth, meaning it took about 10 minutes for it to start working, for it to start killing all the cells. And if you have a clear tube, that means it killed all the cells. If you get one bacteria cell in a broth tube, it's going to grow overnight and be cloudy. Of course, the Bacillus, being a spore former, is hard to kill. So this is an example of a chemical experiment. Please be able to interpret these tubes. What does that mean? What does the 2 cloudy here and clear here mean? These 2 have growth. This one does not. If you had growth in all 4, that means that chemical is not very good at killing that microbe. If in the case of Clorox, they're all clear like this, Clorox is very efficient at killing bacteria. Okay, and that is everything we're going to cover with physical and chemical control. So thanks for joining me.